Hello friends, welcome to BMH Learning. This video is regarding RNAse protection assay. RNAse protection assay is a technique used to detect individual RNA molecules in a heterogeneous RNA sample mixture extracted from cells. The importance of this method is that one or more RNA molecules of known sequence can be identified even at low concentration of sample RNA. Principle. The principle relies on the concept of hybridization. As we know, the two strands of double-stranded DNA are complementary to each other. In a similar manner, scientists can engineer a strand of DNA or RNA such that the engineered DNA or RNA strand complement a specific portion of the mRNA transcript. Let's suppose there is a known mRNA transcript which codes for a particular protein. So, if the mRNA sequence is known, we can design a DNA or RNA sequence that is complementary to that mRNA stretch which codes for the protein. Now, this engineered strand base pairs with the complementary target sequence in the mRNA. This process is called hybridization. So, at the end, we are gonna detect the engineered strand. So for detection, we label it with something which makes it different from the rest of the molecules. Once the strand gets labeled, it's referred as probe. Now, the probe is hybridized to the target mRNA. Notice clearly, we are getting a partially double-stranded complex. Now we are going to treat this complex with RNAse. So, unbound single-stranded RNA is going to be digested or chopped by RNAse to ribonucleotides, whereas double-stranded RNA complex is not digested by RNAse. So, the probe actually protected the mRNA from the action of RNAse. Hence, the technique is named RNAse protection assay. Thus, the newly formed double-stranded RNA complex is resistant to digestion by RNAse and therefore can be identified by high-resolution denaturing polyacrylamide gel electrophoresis. Applications. Nuclease protection assays are used for the mapping of introns and 5' and 3' ends of transcribed gene regions. Amount of the RNA of interest present in the original cellular extract can be detected. So, in this case, if the target is a messenger RNA like we discussed, this can indicate the level of transcription of that specific gene in the cell. It's also used to identify the presence of double-stranded RNA. And if the double-stranded RNA is found, this could mean RNA interference. This was all. Thanks for watching.